Okay, we already looked at how the transgenic animals are going to be formed. Okay, so now we can look at what are the applications of these transgenic animals. So it has different. It is in the PDF form. Oh, sorry. That is the top is transgenic animals. So that the first one, the improving milk production and the composition. Okay, so that the quantity of the milk produced should be increased. At the same time, the composition. You know that in the milk we have different components like the protein, casein, lactose, and other components. Okay, milk, lipid, etc., etc. So that we can the transgenic production, transgenic animals should be formed to increase the milk production and the composition. Okay, so that you know the advances in the recombinant DNA technology have provided the opportunity either to change the composition of the milk or to produce entirely novel proteins in milk. Okay, so that what you can do, you can change the composition of the milk. Okay, either to change the composition of the milk, so increasing some proteins, increasing the lactose, or some sort of thing. So otherwise, there's a possibility to produce entirely novel protein in milk. So if the protein is not already existing in that particular milk, so you want to use the new protein to be produced or expressed in the milk. Okay. So these changes may add value to increase the potential juices of the milk. Okay, so that if you want to reduce the fat content and if you want to increase the protein content and particular component of the protein content, or you want to introduce a new protein into the milk, all these are possible. So that these changes may add value to increase the potential uses of the milk. And the improvement of the livestock growth or the survival through the modification of the milk composition requires the production of the transgenic animals that produce a great quantity of milk and produce milk of high nutrient content or produce milk that contain beneficial protein. Okay, so that you can produce the transgenic animal just to increase the quantity of the milk so that the per yield cow is going to be introduced, increased. For example, we have the indigenous animals with the 1 or 1.5 liters per milk. So that if there's a possibility to produce the transfer the genes into the indigenous animals genome, that will lead to high milk production, then the quantity of the milk produced is going to be increased. Okay. Or otherwise, you can produce milk of high nutrient content, so that the availability for the neonates or the young calves also going to be increased, so that their survivability and the productivity during his lifetime also going to be increased. Or otherwise, to produce milk that contains that beneficial proteins also could be obtained through this transgenic process. So the transgenic alteration of the milk composition has the potential to enhance the production of the certain proteins and of growth factors that are deficient in milk. So that maybe some proteins or growth factors that are deficient in milk. Okay, so the increased expression of a number of these proteins in milk may improve growth, development, health, and survivability of the developing offspring. Okay, so if there is some deficiency, and through this transgenic process, you can introduce the gene that is responsible for the deficient protein or some other factors, so that ultimately the growth, development, health, and survivability of the developing offspring is going to be increased. So some of these factors are insulin-like growth factors and epidermal growth factors and transforming growth factor beta and lactoferrin. So these all have some effect on the growth, development, health, and survivability of the offspring. So other properties of milk that we are consideration for the modification are those of that affect human and animal health. So that then again, if you look at these other properties of the milk that bear consideration for modifications are those that affect the human and the animal health. So that it has been shown that specific antibodies can be produced in genetically engineered animals. And you might be knowing the antibodies, so they will fight against the antigens. So that through this transgenic process, you can allow the animal to produce the specific antibodies against a particular disease antigens, so that we can overcome the health problems to the animals. And another example is to increase the proteins that have physiological roles within the mammary gland itself, so that the mammary gland has its own physiological functions, so that you can increase the protein that have influence, or that influences particular physiological roles within the mammary gland itself, so that the physiology is going to be enhanced. Then the milk production or other properties also can be sort of altered. So that is the first one, improved milk production and composition. So that you can increase the milk production, you can change the composition, you can introduce new proteins or new components to the new gene transfer, and they are going to have the influence on the development, growth, health, and productivity of the offspring. At the same time, you can enhance the animal to produce certain proteins, so the antibodies to help for the animal health and human health as well. Okay. So that the next one is the improvement in disease resistance. So that the disease resistance also one of the factors that influence the livestock production. So when the animals are susceptible to the diseases, particularly when you go for the breeding program or the cross-breeding program with the temperate animals, the disease resistant ability is going to be reduced for the crossbred animals. Okay. So that the improvement in disease resistance, so the genetic modification of the livestock by transgenesis will produce healthy animals, and the healthy animals are an asset to anyone involved in the rearing of livestock. Okay. And if you know that in Tamil they say no yet a valve private seldom. Okay, so if there's no disease, that is a great wealth. Okay, so that is applicable to the animals too. So the healthy animals are an asset to anyone involved in the rearing of the livestock. So thus the technique of animal biotechnology should be more emphasis on producing animals with higher resistance to diseases. So now we are going to introduce the genes which give resistance to a number of diseases to the given species or the breed of the animals. So that the examples are given here. Okay, so yesterday also I was asking the global perspectives. I listed so many case studies. None of you remember those case studies. Okay. But here also for the disease resistance, they have listed some of the examples. For example, mastitis. So the mastitis is a disease condition for the milk producing animal. So the inflammation of the udder, particularly through the unhygienic condition or infection of the pathogens, mainly bacteria. Other organisms also involved, but mostly by the bacteria. So the mastitis is a condition developed due to infection of the udder tissue by a pathogen that results in inflammation like symptoms like swelling, redness, warmth, and the udder pain. So that these are the signs of mastitis. You must be knowing that one. So this results in low milk production in affected cattle. So the mastitis will lead to low milk production. So again, this mastitis milk is not suitable for consumption as well. And it might, in chronic cases, it might lead to the loss of some of the number of teats as well, so that the milk production is going to be permanently affected. Okay, so the results in low milk production in the affected cattle. So the occurrence of this disease can be reduced by producing transgenic cows that carry gene for lycostabine, Okay, so lycostaphin, and secrete the protein in their milk. And what is the role of this lycostaphin? Protein provides higher resistance to mastitis by killing the bacteria. So it is a protein. Okay, so you insert the gene that is responsible for the protein lycostaphin. So this protein has the ability to kill the bacteria which causes this mastitis. Okay. So the lycostaphin protein provides higher resistance to mastitis by killing the bacteria, Staphylococcus aureus. So that is the main one that lead to this mastitis. However, the resistance offered is dose dependent. So lycostaphin is an antimicrobial peptide that protects the mammary gland against this major mastitis causing pathogen. So you don't need to write all these things. You can give mastitis lycostaphin, killing the bacteria, Staphylococcus aureus. Okay. So the third factor is the reproductive fitness. So you know the reproduction also important as far as the animals are concerned to increase the number of animals. And it's not just increasing the number of animals, you want to increase the animals genetically superior animals. Okay. So the improvement in reproductive fitness of healthy livestock is always a positive signal for people associated with livestock rearing, and such transgenic animals producing desired products or exhibiting superior characters are more suitable to meet the requirement of the human population, be it food or therapeutics. So the numerous potential genes have been identified that can be greatly affect the litter size. Okay, so the reproduction means you want to get more number of genetically superior animals. Even after the gene transfer, you want to get more number of that particular animals. So that numerous potential genes have been identified that can greatly affect the litter size. You know what is litter size? So that an engineered estrogen receptor gene can be introduced in the genome of transgenic cattle that can increase the litter size. Okay, so that you are going to introduce an engineered estrogen receptor gene. So now estrogen is one of the reproductive hormones. So that the estrogen receptor gene can be introduced in the genome of transgenic cattle. So that can increase the litter size. For example, introduction of this gene in numerous breeds of pigs effectively increases the number of progeny and thus the availability of the food as. Okay, so when the number of progeny is increased, so that you know the pigs are mainly reared for the fattening purpose, so that the food availability also going to be increased. So that is indirectly or directly it is going to contribute to the food security for the human being. And the second example is associated with the reproductive fitness is a gene for the fecundity has been identified and called as the Brula fecundity gene. And in merino sheep, which allows for increased ovulation rate. Okay, so the Brula fecundity gene, fecundity you have studied under the biology as well. Okay, so increasing the fertility. Okay, so Brula fecundity gene in merino sheep, which allows for the increased ovulation rate. So that when the ovulation rate is increased, then the conception rate and the number of offspring you are going to get also increased. So the gene increases the ovulation rate by approximately 1.5 over, so that the transgenic sheep carry the fecundity B gene can be produced, which will increase the fecundity in different breeds of the sheep. Okay, so that if you want to increase the number of ova sheep as a result of the number of offspring, you are going to obtain this this gene has already been identified in the merino sheep. So this gene could be transferred to the other sheeps where it can increase the ovulation rate by 1.5%. As a result, the reproductive rate of the other sheep also can be increased. And next is the woolen hair fiber. And you know the wool you get particularly from sheep, and the hair and fiber maybe from the other animals like some of the goat beefs and some of the sheep beefs also might give this one. So the harvesting of the wool from animals like sheep is of economical significance the world over. So the qualities that are important in wool include the fiber quality, color, yield, and the ease of harvest. So the animals can be manipulated for these desired traits easily by employing transgenic techniques. Okay, so you want the wool fiber quality can be modified through the transgene, color can be modified, yield can be increased, and the ease of harvest also could be done by employing this transgenic technology. So the quality, the strength, the fineness, length, and crease of hair fibers and wool fibers from sheep and goats have already been manipulated. So all these parameters associated with this wool, so the wool, hair, and fiber has been manipulated using this transgenic technology. So that the future prospects will focus on the surface of fibers, wherein the surface interaction can be decreased by genetic engineering. So that now what they want to focus is the surface interaction should be decreased by genetic engineering. So that will result in decreased shrinking of the garments made from these modified fibers. Okay, so that due to this surface interaction, the garments are shrinking. So using this gene technology or transgenic technology, they want to introduce some genes so that will reduce the surface interaction. As a result, the shrinkage of the garments can be decreased. A recent approach has been to produce a spider cell which has tensile in strength. This silk is a highly useful fiber and can be secreted in the milk of the transgenic goats. Okay, so you have this spider silk, so that is secreted by the spider. Okay, and that which has a high tensile strength, so you can stretch it for a longer length. So the silk is highly useful fiber and can be secreted in the milk of the transgenic goat. Okay, so this spider thing was the gene was transferred to the goats, and this gene was expressed in this transgenic goat's milk, and from the milk this one is separated, this protein is separated, and this is sprayed over the clothes or something, so that it provides the high tensile property to that. So it can be elongated up to 35% and has tensile properties similar to the synthetic fiber Calva. So the energy absorbing capabilities of the drag-line silk exceed those of the steel. Okay, so that this protein, the drag-line silk protein, has the desirable properties that exceed those of the steel. Okay, so that they allow the goat to produce this particular protein in its milk, and the goat milk will be obtained, and the protein from this milk will be extracted and utilized for the manufacturing purpose. So because of its properties, drag-line silk finds many potential use like in medical devices, shoes, ballistic protection, tire cord, airbags, aircraft, automotive, composites, and clothing, etc. So it's not only for the clothing. You can see this protein has several uses, so that the goats are produced where in the milk this particular protein will be excreted. So then it will be extracted from the rest of the component and utilized as the spider cell. It has several uses. And next is the breeding of the transgenic animals. And traditionally farmers have used selective breeding, that you know. Okay, so based on the phenotypic characteristics, they select the animals and they breed, and they expect the phenotypic performance. Okay, and we had some ways to estimate the breeding value as well based on the genetic relationship and the phenotypic performance, etc. So that traditionally farmers have used selective breeding to produce animals so that exhibit desired traits like increased milk production, high growth rate, increased resistance to diseases, etc. However, these methods are time consuming. Okay, so the traditional or conventional method of selection is time consuming. It takes so many generations to get a given rate of improvement. And again, through the normal selection process, the genetic improvement we can make is only 1 to 2% per annum. So you could see how many years it will take to increase the performance level to a higher level. Okay. So the time consuming and difficult. So when the technology utilizing molecular technique was developed, it became possible to develop desired traits in animals in a short span of time, and with much more precision. Okay, so that okay, you know, okay, this is the gene responsible for this protein or this level of production. I will extract that gene and I will transfer that one to another needed animal so that the production will be increased or the composition will be modified, sort of. Okay, so that moreover, it is easier for the farmers to increase the yield using these techniques, be it food or secreted proteins. Okay, so that it can increase the yield and it can be served as a food to farmer. It can, how do I say, can serve as a food to meet the food security or otherwise particular protein that is needed for the human being. Okay. And the other one is the medical applications. So the xenotransplantation, so that is the organ transplantation. So the organ failures in most of the patients require transplantation of the organ. So the progress in the medical field regarding transplantation technology has led to more successful transplantations. And this has led to the acute shortage of the suitable organs. Okay, so nowadays you could see the kidney transplantation, that and this heart transplantation. So this under the leadership of this doctor, this operation was successful like that. Okay. So with the advancement of the medicals, the organ transplantation also become a suitable, feasible area where the transplantations can be done. And to meet this increase in the demand of the donor organs, porcine xenografts are considered as an alternative. So porcines are the swines. Okay? So swine xenografts, the organs are considered as an alternative. Pigs are considered suitable as the organ donors for the humans because their organs are similar in size to human, and they produce a more number of progenies, and the progenies mature early. Okay, so that they have the similar size to the human being organs, and they produce more number of progenies, and the progenies are mature early. So that from a group of organs, if you are needing a number of organs within a shorter period, you can get it from the swine. Okay, that is meant by the more number of progeny and the early maturity. Consequently, pigs can be genetically modified to become viable animal source for the tissue and organs for the human transplant. So that due to its other above mentioned factors, similar size of the organs to the human being and more number of progenies and early maturity. So pigs can be genetically modified to become viable animal source for tissues and organs for human transplants. So that the modification includes the deletions of the genes responsible for the human immune rejection response. Okay, so now you see, if you are transplanting that the pig organ to the human being, the animal body will consider this one as a foreign substance or foreign organ, so that the immune system will produce antibody to reject this one. Okay, so that is meant by the human immune rejection response. So such modified pigs are known as the knockout pigs. Okay, so that the modification includes deletion of the genes responsible for the human immune rejection response. So that is the gene that's going to produce some antigens. So that will induce the human body to produce the antibody to reject that one. Okay? So that the genes responsible for the rejections are going to be deleted from the pig organ, so that the rejection response won't be there, so that there will be a compatibility. So they are otherwise incompatible. Okay, here compatibility is going to be increased. So there are such modified pigs are known as a knockout pigs, or the knockout pigs in activation of particular gene. So they can either be cloned, or their fecundity rate can be increased to maintain a steady supply of the animal source for the xenotransplantation. So these all are the possibilities. Okay, these all are the possibilities. So you scientists can think of it. Okay, however, such transplantations are not easily acceptable by the general public due to many social, ethical, and religious sentiments. Okay, how come I have an organ from the pig? Okay, so that is social. Okay, or otherwise this man is having some organ from the pig. Okay, that is social. So ethical, so we already looked at the ethical, the species integrity is sort of overlooked, or how do I say, overruled. Okay, so the ethical issues also there. And the religious sentiments, for example, the Muslim community, they don't like these pigs at all. Okay, due to their religious reason. Okay, so for them, the transfer of organs from the pig will not be possible at all. So those are the religious sentiments. Okay, so the social, ethical, and religious sentiments are there. And next is the blood replacement. So presently the products from blood needed by the hospitals are produced or extracted from the donated blood. However, this is quite cumbersome and also limiting due to lack of qualified donors, disease concerns, and other regulatory issues. So a possible solution of this problem is the production of transgenic animals that carry human antibody genes, so that can produce human polyclonal antibodies for treatment of various diseases or conditions like organ transplant rejection, cancer, and autoimmune diseases. So that you want to produce the human antibodies through the transgenic animals. Okay. So the polyclonal and monoclonal, that also you should have studied. Polyclonal antibody or the monoclonal antibody production, you have as one of the titles on the market. Okay? It's not for me. Okay, so the possible solution of this problem is the production of the transgenic animals that carry human antibody genes that can produce human polyclonal antibodies for treatment of the various diseases or conditions like organ transplant rejection, cancer, and autoimmune diseases. I already taught you what is this organ rejection. And the transgenic pigs can be produced that express human hemoglobin for use as a blood substitute. Okay, so that hemoglobin is the main component of the blood. So that transgenic pigs can be produced that express human hemoglobin. Okay, so that for the use as the blood substitute. Okay. So that again the hemoglobin can be extracted and purified from the pig red blood cells, and the explanation for something is given here. And next is the production of the pharmaceuticals from the transgenic animals. So the pharmaceuticals means the drugs for different treatments. Okay. So several recombinant proteins of pharmaceutical significance can be made to be secreted in the milk of transgenic cattle. So the gene for the product can be introduced in the DNA of the animal, and the resultant transgenic individuals can be screened, and the population of the transgenic animals can be maintained. And you know that when you transfer the gene, not all the animals are going to carry the gene, so that you have to identify the animals which are carrying this transgenic genes, so that you will screen the animals for the presence of the particular transgenic gene. Then you will multiply those animals to a higher number, so they will express that particular gene. So the transgenic animals are the source of several recombinant proteins like enzymes, blood clotting factors, vaccines, monoclonal antibodies, growth factors, collagen, fibrinogen, so that we can use many of these recombinant proteins that is produced in the recombinant genes. So that most transgenic species are studied for research applications. However, many are used to meet the demand for commercial and pharmaceutical sectors. Okay, so that so these transgenic species can be used for the research purpose and to meet the demand for the commercial and the pharmaceutical sectors as well. So this table below gives some of the transgenic animals and the products that can be obtained from them. Okay, so just you can go through it. So the next one is the as a disease model. So the historically mice have been used to model human disease because of their physiological, anatomical, and genomic similarities to the humans. And transgenic animals are produced so that use the animals as a model to human and study the development of the diseases or the action of the different drugs, etc. So transgenic animals are produced as a disease model. So animals genetically manipulated to exhibit disease symptoms so that effective treatment can be studied, such as Alzheimer's, cancers, and AIDS. Okay, so artificially the disease will be developed into a set of animals, and different drugs will be utilized to test its effects. And the transgenic animals enable scientists to understand the role of the gene in specific diseases. So the benefits of using transgenic animals include the possibility of the replacement of the higher species by lower species. So through development of the disease model in mice rather than dogs or non-human primates. Okay, so that they want to use the lower species than the higher level of species like dog and primates. So the extent of discomfort explained by parent animals during the experimental procedure. So that again, okay, they want to make use of the animals to study the human disease development and the functions of different drugs to control the disease. But even within that, they have some ethics. So to use of these lower species and not the higher species. Okay. So that the transgenic animals such as mice have been found to be valuable in investigations into gene function and for analysis of the different hereditary diseases as well. So these the transgenic animals for the environmental use. So here, for example, this the EnviroPic I already would have told you. Okay. So that what we have here, the pig is a monogastric animal. So when you feed the diet with the high level of phosphorus, these animals can't digest this phosphorus and make use of it. So that high level of phosphorus is going to be excreted in its species, and the environment is going to be loaded with the high level of phosphorus. Okay. So under such a condition, if you could allow this animal to excrete this phytase enzyme in its saliva, then it can easily digest these phosphate, the phytic compounds, and the phosphate can be digested, and the phosphorus is going to be absorbed by the animal. So that's the load, the environmental dose or load of the phosphorus is going to be reduced, so that the production of the pig with the enzyme phytase, which is secreted in the saliva. So that is known as the EnviroPic. Okay. So likewise, there are some examples in the fish as well. Okay. And at those things, so these are the applications of the transgenic. Okay. Application. So this one also you don't need to write all these paras. You just pick up the main points. Okay. So now we are going to move on to